So it's a pleasure to introduce Kurt, who was my thesis advisor. So Kurt is famous for many beautiful results in theoretical physics. One of these was the Callan semantic equation, in which he explained how physics at one length scale relates to physics at another length scale, and other length scales, a very important kind of topic in physics. Another famous result of Kurt's is the Callan-Rubikov effect, which explains how the existence of magnetic monopoles, if they exist, can catalyze the decay of the proton. Besides that, there's many other things that Kurt has done, too many to indeed name, and he's the recipient of many famous prizes. So I'll tell you that one of the things that sort of made me, that impressed me most about Kurt when I was his graduate student was his sheer erudition. He seemed to know everything from art and history and sophisticated mathematics and everything else. So true to form, in recent years, Kurt has been working at the interface of physics and biology. And one of his most important interests in that area has been basically the dynamics of the adaptive immune system that vertebrates like us use to fend off, all kinds of bugs that want to otherwise do us harm. But today, Kurt is going to give us a historical talk about Richard Feynman, who was one of the seminal figures of 20th century physics, famous for all manner of things. And apparently, he was also very interested in problems in biology, a thing I didn't know. So Kurt's going to tell us about Feynman's forays into biology and what lessons we can take from that. Let's welcome Kurt Callan.
Well, if it's any comfort, I'm much less erudite today than when you were a graduate student. Maybe you just gradually realize as you get older just how little you know.
When I first walked into your office, on your table, there was the journal Foreign Affairs, which I've never seen any other physicist's table.
Yeah, don't I have trouble enough? Okay, so I think this is going to be a somewhat unusual lecture from the standards of this lecture series. Vijay, when he asked me to give a public lecture when I was here for doing my thing at the center, said they wanted to have at least one per summer talk of a philosophical or a cultural or a historical nature. And as it turned out, I had something more or less in the can roughly of that description, in the sense that I had the good fortune to participate in a conference in Singapore back last fall to celebrate the 100th anniversary. Something's wrong. No? We're okay? All right. The 100th anniversary of Richard Feynman's birth. He was born in 1918, and so he was the age of my father, roughly speaking. And it was kind of interesting. A bunch of people who had either studied with or worked with Feynman over the years were there, and it was a real nostalgia fest in many ways. I had never worked with Feynman. I was not his student. I never collaborated with him. I did spend a little bit of time at Caltech, but Feynman was notoriously reclusive when he wasn't being outgoing, and I guess he was in a reclusive mode when I was there. I did have one interaction with him. He came to give a lecture like this at my college, which was Haverford College in Pennsylvania. And he gave a kind of general public lecture, which was his usual bravura performance. And then he gave a talk to the people who were physics majors, and there were about 5 of us. And then we must have dragged in a few chemistry majors as well. And he gave a talk which was a kind of souped up and compressed version of one of his sort of standard big audience talks on quantum electrodynamics. And I was put up by my professors to ask a question at the end. He was famous for something called Feynman diagrams. And I'm not going to talk about Feynman the physicist very much, but this was at the core of his achievement in physics. And somehow there are things you can scratch down as little sketches on a piece of paper that somehow solve a complicated physics problem. Let's put it that way. And so I was put up to ask the question, could you explain to us, Professor Feynman, in terms that we might hope to understand, what are Feynman diagrams? And he this was a bit of a problem. he gathered himself together. No, he didn't. He sort of, right off the bat, he said, "Kid, if you could understand the answer, you wouldn't ask the question." So he could be a little prickly as well. Okay, so I'm assuming that most of the people in the audience have heard of this gentleman, right? I mean, he's not as famous as Stephen Hawking, but in his day, he was as famous as Stephen Hawking. And for reasons that I will get into, he was a good client of the, I don't know, the literary and the, I don't know, the biographical industry. There was not just his physics to talk about. So let me just set the stage a little bit. So Feynman was one of the great theoretical physicists of the 20th century, and I'm not going to be able to explain much about that. But he had a flamboyant, colorful persona that made him interesting to audiences that really knew nothing about his physics. And I gathered together a couple of photographs here which say something about this. He was a curious character, deeply and actively interested in science outside physics and in many areas far outside science. I'm going to focus in this talk on his adventures in biology, which I think virtually none of my colleagues in physics know anything about. And I didn't know anything about until I was given the short straw at this, I pulled the short straw at this conference on Feynman, his 100th anniversary of birth. There was something left over, Feynman this in physics, Feynman that in physics, and Feynman something else. Feynman is a social figure or something. You figure it out. I got the short straw. And since I'm interested in biology, I just went after the biological side, and I found a few things, and I found it quite interesting. So here's some pictures, just to put some flesh on the bones here. So this is Richard sitting in front of a blackboard, and I suspect he's talking about liquid helium. And here's Richard in a, I don't know who created this painting of Richard as a wizard. He was a wizard. He was a theoretical physics wizard. He's the kind of person whom, as a theoretical physicist, you say, I don't care how smart I am. I could never have the kind of insights that he had, just coming, tripping off the tongue effortlessly. However, I should point out, just to give you some sense of how this guy interacted with the world, this painting is in a mosaic on the back wall of a Ross Dress for Less in Pasadena, California. It's multiple life-size. I'm sure it exists as a painting in somebody's house, I hope, but it was reproduced in Pasadena, California, where he spent most of his scientific life, facing a parking lot where a couple of big box stores live. Okay, so this is something, tells you something about the social impact or the beyond physics impact that this guy could have. Now, he had two very important passions outside of physics. One was music, if you call bongo playing music. And apparently these aren't bongos. I'm not sure exactly what they are. But he learned to play this kind of drum, and he played them in public for friends, for relaxation and amusement throughout his life. And also, this charcoal drawing of the head of a woman, head of a young woman, is hanging in my office. Somehow, I don't know why, after his death, a large collection, at least a dozen of drawings and even one painting in watercolor showed up at Princeton University Physics Department. And we didn't quite know what to do with them. And we've distributed them around amongst our colleagues. And so he got interested in art, in painting and drawing. And he found in Pasadena an artist by the name of Zorthian, who, I guess, Zorthian came to him, or he interacted with him in some context. And Zorthian said, I want to learn about physics. Could you tell me about physics? And Feynman says, well, if you'll teach me about drawing, I'll teach you about physics. And so every Sunday, Feynman would go over to Zorthian's studio, and he would talk to him about physics, and Feynman would get some lessons from him about drawing. And this is not bad. This is actually a pretty competent piece of art, it seems to me. So he was a curious character, interested in many things, and in particular, he was interested as I'll explain, for a time in biology. Okay, so I had to give a historical talk, and so if you do something historical, you have to have sources. So I rooted around, and there are several sources that I dug into, and you know, this is a secondary source, the book. Surely you're joking, Mr. Feynman. Many people in the audience probably have actually read this book. And you will understand when I say that he was a colorful, flamboyant character. He liked to perform, he liked to show off, and he could very, very well. There's another one that I'm going to come to called There's Plenty of Room at the Bottom. This was a talk he gave that in 1959 that was actually quite influential, and this was published in, you know, the alumni magazine of Caltech. And then there is his famous lectures on physics, which have here and there some biological tidbits. And then later in the 60s, I'll give the timeline in just a second, he actually gave a very extended series of lectures to the engineers at Hughes Aircraft on modern science. He did physics, he did chemistry, and he did biology. And so these things are available online. He didn't actually write this up, somebody in the audience did. So I read with some care his Hughes lectures on biology, organic chemistry, and microbiology. And then there's one paper that he wrote with, he was a postdoc of a postdoc, as it were, in a laboratory in Caltech. And I'll say something about this paper, which is the one published bit that came out. And then there was a paper that actually drove me in this direction that was published recently, When Physics Meets Biology, a less known Feynman. Okay, so Feynman's adventures in biology, a timeline. So he was a graduate student in physics at Princeton University from 1939 to 1942. His thesis advisor was a gentleman named John Wheeler, whom I knew very well, who was in his own way as, you know, off the median as Feynman was. And between them, they did 2 or 3 really quite remarkable things. And Feynman went off after he got his degree in 42 to Los Alamos, where he was part of the team of physicists who built the bomb. And in his Surely You're Joking, Mr. Feynman, I think there are a bunch of anecdotes about hijinks at Los Alamos, you know, doing things to torment the security officers and things like that. And then after the end of the war, he did a series of works that basically crystallized modern quantum field theory. He was not the only person, but he did a very remarkable thing in understanding how to make the physics of photons and electrons, what we call quantum electrodynamics, a fully defined and manageable and computable theory of what really goes on in the world. He got the Nobel Prize in 1965 along with Julian Schwinger and Shinichiro Tomonaga for what he did in the late 40s, basically. So he eventually landed in the early 50s at Caltech as a professor of physics. I probably had some fancy title even at that young age. This happened to be, however, the 50s, one of the 2 or 3 most central places in the world for the creation of modern biology, what we call molecular biology, the understanding of biology as the coming together of some very specific and important molecules, DNA, RNA, and things like that. And the leader of this activity at Caltech was a physicist by the name of Max Delbruck, who had gone full native. He had an experimental lab. And Feynman exercises his curiosity, which he'd begin to exercise at Princeton as a graduate student. He took a couple of courses in biology and enraged the other biology students with the kind of questions that he would ask. But when he got to Caltech, he would hang out in Delbruck's lab from time to time. He met and interacted with and impressed with his incisive insights, current and future leaders of the new field of molecular biology. I'm not sure what happens in between, but I think this interest in biology, just as a kind of entertainment or exercise of his physical intuition and mathematical insights, continued for a while. In 1959, he gave this talk, Plenty of Room at the Bottom, which I will go into in some detail. And in this talk, he challenges physics to go to work at molecular scales, imitating life. In this talk, he foresaw the miniaturization revolution in computing and espoused a physics-inspired view of the core issues of biology. And this was just a talk he gave to the Alumni Association, and he wrote it up, or somebody wrote it up, and it is a kind of foundational document for many of the things that we take for granted in our electronic miniaturization era. I don't know if the two things are connected, but in 61, 62, he had a sabbatical, and he decided to take it in place working at the bench in Delbruck's lab, and he studied reverting mutants of bacteriophages. I will see if I can explain what that is. And it led to a serious paper in the journal called Genetics, and further work with Messelson and Lamson. Messelson was a graduate student at that time. He became one of the giants of modern biology. He worked with them on ribosomes, and he did work that targeted core issues of the new biology, but somehow, because he only did this for the restricted period of time that he was on sabbatical, somehow, you know, you really have to dig in when you're doing something that involves experiment. You can't mess with it. You either get it right or you don't, and getting it right means just slaving over detail after detail after detail, and that wasn't his thing, really. In 62, 64, the Feynman Lectures on Physics had one biological chapter in color vision, and if I have time, I will extract something from that, which I think is interesting in its way that it shows how physics and biology can talk together in a useful way. Yeah, and in an aside, he showed that the insect eye optimizes its function subject to the constraints of physics, and this foreshadowed a major trend. And then there were these Hughes aerospace lectures on biology and chemistry. They were pedagogical, but they contained Feynman's evolved view of the core problems of biology and some thoughts about how physics and biology should interact. And I will come back to that as sort of the leaping off point for what I want to say about what lessons we might hope to draw from Feynman. So first, a little bit of stuff. Okay, this is a complicated slide, but I just wanted to riff a little bit on what was known about molecular biology by the late 1950s or early 60s. A lot was known, but an awful lot wasn't known. I mean, I was surprised actually to sort of reboot my knowledge of the history of molecular biology. The structure of DNA was known, but it wasn't known in any detail how it was replicated. The structure, as Crick and Watson said in their paper, it has not escaped our notice that this structure clearly must be closely connected or could be used as the foundation for understanding how the accurate genetic code is transmitted from one generation to the next, because it's complementary base pairing. But exactly how you do it without getting all tangled up and what enzyme does it was not known. And the other thing is, we now know, if you know anything about modern biology, you know that DNA is a sequence of these bases, A, C, T, and G, adenine, cytosine, thymine, and guanine. But what actually gets pumped out is not bases, but amino acids that string together to make proteins. And the way in which the proteins were made was not known. It was known that there was a thing called the ribosome, which somehow translated the code for proteins that was contained in the sequence of bases in the DNA, but exactly what was going on was not known. But what I wanted to say was the thing that was most not known was, okay, how do you connect between the sequence of bases in the DNA and the amino acids that get strung together to make proteins? There are 20 of them, and there are only 4 bases, so there's got to be some kind of translation. And it was discovered, you know, in the early 60s that it was exactly a triplet code. That is to say, every 3 bases would code for a specific one of the 20 amino acids with with 4 bases, and if you put 3 of them together, you could in principle code for 64 different things. So it's a degenerate code and so on and so on. But this was all up in the air, and this was up in the air at the moment that Feynman was actually working at the bench. And what he did working at the bench was not unrelated to the very soon thereafter recognition of exactly how the genetic code worked. Okay, so this is a tad technical, and I'm not sure how far I'm going to be able to go into it, but just to give you a brief idea of what was going on in the lab of the real biologist, the real biophysicist, Max Delbruck, it was known that DNA existed. It was known that DNA coded for the organism itself. It was kind of known that the DNA coded for specific proteins. And it was also known that this code was linear. That is to say, the DNA was a long molecule, and somehow as you walked along this long molecule, you would run into information that had to do with first this protein, and then this next protein, and then the next one. And the challenge was to find out, well, where could we in fact map out on this linear structure? Could we map out where this protein is, where that protein is? Could we sort of say, make a linear map of where the proteins that are essential for that really make up the structure of the organism are to be found on the DNA? So Delbruck and some other people had the clever idea of using viruses that infect bacteria. So the viruses that infect bacteria are very, very small viruses. Their DNA is very short and only codes for a few proteins. This was vaguely known. The idea that you have a problem of finding out where 5 proteins live on some stretch of DNA is much more likely to be manageable than figuring out where 20,000 proteins are coded for on, let's say, our DNA. And basically, they did it by isolating mutants of a particular strain. And they could tell that this virus is different from this virus by seeing what happened when this virus infected a standard bacterium, E. coli. And so basically, if you have a Petri dish with a lawn of bacteria on it, and you drop a few of these viruses onto this lawn, the viruses will be taken up by the bacteria. The bacteria will, the point of the virus is it hijacks the machinery of the cell, and it causes the cell to make many, many, many copies of the virus, the single virus that's infected it, and kill the cell, explode, and release into the surrounding region a lot of new virus particles. And you can tell by looking at the little spot on this Petri dish, you'll get different kinds of spots. Some will be smooth, some will be wrinkly, some will be colored, I don't know what. But the biologists were able to identify different actors. So you could tell that this virus was this kind and this other virus was a little bit different. And then basically what they did was to co-infect. That is to say, they looked at what would happen when you infected essentially the same bacterium with 2 viruses simultaneously. Then their DNA is all mixed up inside the virus, and it turns out that this was a gift of nature, that if the DNA of the viruses that are different by some small number of mutations, it's allowed to come together, then it just naturally engages in miscegenation. That is to say, here are two strands of DNA. This one is a little bit different from that one. It has a mutation here, and this guy's got a mutation there. But other than that, they're very similar. Well, they can exchange. That is to say, the first half of this guy can go together with the last half of that guy. And so you can cause these mutations either to be shared by some new piece of DNA or eliminated entirely. Okay? In other words, you can basically cross together 2 such strings of DNA and eliminate the parts that have the mutation. And then you would see, then you look for those interesting crosses by essentially collecting these viral particles that are made by the infection and infecting a new set of bacteria and looking for the original strain, the original, phenotype, a nice clean spot on the Petri dish. And how frequently that would happen, this crossing over and eliminating the mutations, how frequently that would happen, that would tell you something about how far apart on the DNA the mutations were. So they use this kind of rather painful experimentation to map out where different genes, or rather where different mutations were, but these were known to be mutations in different genes on the phage DNA. Okay, so what Feynman did was, he engaged in a hunt for more such mutations that could be caused to be undone by essentially causing co-infection of a bacteria with two separate mutations. He found he and his team found some mutants that were interesting, and 2 years later it was revealed that the piece of evidence that he had found was actually evidence for the base 3 genetic code. But it wasn't really understood at the time. So there was a paper in 1961 called The General Nature of the Genetic Code for Proteins, and this was the paper in which Crick and I don't remember the name of the other authors of this foundational paper, it's my problem. But basically this is where the first realization that the code that was instantiated in the DNA was a triplet code, that every 3 bases corresponded to an amino acid. So basically what Feynman had stumbled on without realizing what it was that there was a way for a mutation to undo itself, and probably it's not going to be possible to really go through it. But the idea was, if you had some stretch of DNA, the so-called wild type, it's what was there originally, there are various ways in which a mutation can happen. But one of the interesting ways is that a base that wasn't there before can be inserted. So here's the wild type, and I put this guy B in red for graphical reasons that will become apparent. One way you can make a mutation is to just mistakenly insert a base that wasn't there before, an A or a C or a T or a G. I just call it X. This guy was inserted between this B and this C. So what has that done to you? Well, what that's done to you is that you have basically totally screwed up everything downstream from that. In other words, you're reading this sequence of letters 3 by 3 by 3 by 3, and each group of 3 letters corresponds to a particular amino acid. And if you basically shift the reading frame, as they call it, you were fine up to here, and maybe this guy is wrong because you've put this X, which probably isn't the right thing. But in addition, since you're reading it 3 by 3, and you know where you start, you've put everything else downstream in the wrong reading frame. You're going to get stuff that doesn't make any sense. But then one way to undo this, so this is going to be a protein that is totally different from the previous one. Whatever the previous one was doing, this one is not going to do it for possibly 2 reasons. This could be just a sequence, it's a totally different sequence of amino acids. Therefore, it's not going to be the same protein. Why should it have the same function? And in addition, it might even be something that isn't fully realizable, because one of these, two of these triplets actually say, stop translating here, give up. All right, so you can create a stop codon. Well, you could rescue this by, in a second mutational event, if you can insert a base as an error. One of the things that you can do as an error is to remove a base. So suppose, just for the heck of it, you remove this guy, the thing that this B is still here. I've marked it in red so that we can follow it. if you remove it, if you go through the trying to work out exactly how the reading frame works, you'll see that you inserted one and then you deleted one. Well, you know, if this happens close enough together, then there's, although you will have wound up messing up a few of the amino acids in the middle, you will have gone back to whatever the previous wild type string of letters was. All right, so an insertion followed by a deletion, if the two happen close enough together, can in fact leave you with a rescued protein that will work just fine. This is what Feynman actually saw happening. So this phage work that he did came close to something important. It got him an invitation to explain his work to Watson and friends in Cambridge, Massachusetts. But one summer of phage slavery was enough for him. A paper came out. He was working with a postdoc, Dick Edgar, who also became a very distinguished biologist in later years. And the paper was written up. And indeed, this paper, an unpublished work by Richard Feynman, was cited in the 1961 paper by Crick et al. And, but for the rest of his sabbatical year, he focused on other things. And he came close to pulling off a classic experiment using bacterial ribosomes with plant messenger RNA to produce plant proteins. But experiment is hard. And his ribosomes were contaminated in some fashion, so it didn't really work. So, but he learned a lot. He learned a lot about biology. Now, it might have been in the same year, 1961, he delivered this manifesto, Small is Beautiful. So in this article, there's plenty of room at the bottom. He was good with words. In fact, the few things that I've seen that were written in his hand, even letters that he wrote he claimed that he had no sense of social responsibility. He only did what he wanted to. But at this conference, I learned that he actually was a very humane individual, despite his response to me when I asked for an explanation. And I got to see some letters that he had written to people that had just written to him asking for advice. And for some reason, he decided he wanted to write back. They were gems of English expression. So he was good with words. And there's plenty of room at the bottom is one of the classic paper titles or talk titles. It really does say, it draws you in. So I pick out a couple of sentences drawn from this talk. Why cannot we write the entire 24 volumes of the Encyclopedia Britannica on the head of a pin? We can. That is to say, he went through the qualitative physical arguments for why you could do this. In other words, how would you, what would you be doing to write the letters? Well, you would be putting down atoms on some substrate, and you would have to, you could put them down in such a way that the area occupied by a single letter could plausibly be something which was, maybe 10 atoms on a side. He could argue convincingly that it would just sit there. It wouldn't, this would be well written. It would be preserved. It wouldn't evaporate away. And then he computed, well, how many letters are there in the Encyclopedia Britannica, and how much area would this take? And when you're all done, well, a little tiny chip of material would allow you to write the entire content of the Encyclopedia Britannica on the surface of this bit of material. Another thing, a biological system can be exceedingly small. Cells are tiny but very active. They manufacture chemicals. They move and do all kinds of marvelous things. Can we make tiny machines of our own devising? And what if we could miniaturize computers? If they had millions of times as many elements, they could make judgments. Mechanical computers are too big. I want to make submicroscopic computer elements. This was in 1961. Okay, so, and these are threads that were picked up. I mean, they would have been picked up anyway, if independent of Feynman writing down this talk or giving this talk, but it's just an indication of how he thought, as far as thought could take you, given what you knew at a given time. But in this talk is included Feynman's view of the problems posed by biology, and that's what I wanted to focus on. So he says, the most central and fundamental problems of biology today. What is the sequence of bases in the DNA? What happens in a mutation? He'd almost gotten there. How is the base order in the DNA connected to the order of amino acids in the protein? They didn't know in 1961. I couldn't believe it. What is the organization of the ribosomes? The ribosomes are the machines that take the information, the sequential information in the DNA, and turn it into a sequence of amino acids, i.e. a protein. How are proteins synthesized? Where does the RNA go? Now, everybody, all the biologists knew that these were the problems. And he says, from the point of view of the physicist, it is very easy to answer many of these fundamental biological questions. You just look at the thing with a good enough microscope. And he realized that, or he gave the argument that, well, the laws of physics did not exclude being able to actually look at the scale necessary to answer these biological questions. So he, in addition to his challenge to the world of computers, I mean, there were computers at that time, big computers. The IBM 360, I think, could be bought, and it could do some serious computation. But they knew they had to go bigger or smaller. Anyway, he was arguing for smaller. But this simple manifesto for physics coming into biology, I think, is worth noting. I'm checking the time. Okay, so I can't, the clock is obscured for me. Yeah, okay. So I think I'm going to skip over biology and the Feynman lectures in physics, except to tell you that he did a really cute thing, just as a throwaway, to confuse the students. The students were totally confused. It was a set of lectures on all of physics for the freshmen at Caltech. And in addition to being flamboyant and colorful and bongo playing and all the rest of that, he was an extraordinary lecturer. You know, he could convince you that you understood this abstruse thing that you didn't understand before, and while he was saying it, you understood it. That's for sure. You felt you'd been illuminated. And so, and the teaching, the postdocs, the teaching assistants in this course for freshmen were surprised, or they could not understand why the students were often baffled. They couldn't do the problems that were set. And they actually got a taste of their own medicine when the subject became color vision, which is a very complicated thing. And he gave a very clear exposition of how color vision works. And the postdocs and the TAs were as confused as before, as the students, for once. They already, when it came to the elliptical motion, the elliptical orbits of planets around the sun, or the law of the increase of entropy, they already understood that. And to hear it explained in Feynman terms was a real revelation. You felt, now I really understand what I understood. All right, so there's a lovely throwaway that explains that the design of the insect eye is optimized within the limits set by physics. So insects, I guess I'm going to say something about this, but I'll avoid the equations. Insects have a compound eye. So each little part of the retina, each little receptor is at the bottom of a little cone. And these cones are packed together, as you can see. If you've ever seen a blow-up photograph of a fly's eye. So each of those cones is looking at some narrow angle, and at the bottom of the cone there's a nerve ending or there's a photoreceptor basically that says, is there light at this end of this tunnel or not? And so if you want to have more resolution, you'd think, well, you just make these cone angles smaller and smaller and have more of them. Why not? Well, there's diffraction of light. That means eventually, if you make the cone angle too small, it really can't look in a precisely defined direction. So there's a competition between the blurring of diffraction and the desire to have more cones looking in more discrete directions. And there's a best choice in which you make the size of the cone, the diameter of the cone, basically the square root of the wavelength of light times you know, the overall bigness of the eye, the radius of the sphere on which the cones are distributed. And as a matter of fact, actual insects follow that law very nicely. And this fact, and even the explanation, had been noticed by a biologist 10 years before, which Feynman didn't know anything about. So this guy Barlow, Horace Barlow, was one smart cookie of a biologist. I'll say that. Okay, so come the Hughes lectures on biology. Absolutely magisterial overview of the subject. He started with basic chemistry, explained chemical bonding, and then he explained how with carbon and hydrogen and a few other elements you can make organic molecules, and so on and so on and so on. So he says in this sort of introduction to his lectures on biology, to find an appropriate starting point is an impossible job. The subject matter differs from chemistry and physics in some significant ways. All the related fields of biology to a great extent are independent. The reason for this is biology lacks a basic foundation of fundamental laws. Laws developed by theory and proven to be true by experiment. We will find very little mathematics as we go through the material. Basic algebra and calculus will suffice. Lacking this underlying unifying thread, as in physics, I have found it most difficult to organize the material in a meaningful way. So this is in the late-ish 60s. And he knew what we knew at that point, and that's what he says. I've tried to orient the subjects on a scale ranging from the very small to the very large scale where many complex biological systems interact in a closed environment. This is the field of ecology. Thus, I plan sooner or later to span the biological spectrum from the submolecular level to the grandiose ecological level. And it's my plan to start at the bottom and work my way up. He never got to the ecology, but he did actually in maybe 10 lectures go from the constitutive molecules up to some important many cellular things like the functioning of the immune system. Okay, so this led me to ask, how did Feynman's view of biology evolve over the decade between Room at the Bottom, that was 1961, and the Hughes lectures, 1968 or so. So Room at the Bottom is very much inspired by molecular biology, which was a work in progress at the time. He took inspiration from the nanomachines of the cell, and he issues a challenge to physics. Create the tools to see these machines. And he explained while he was doing so why there is no obstacle in principle to do this. He focuses on the molecules that do the cell's work and proposes to understand them essentially by looking at them directly. Totally molecular biological in spirit. The Hughes lectures display a more systems view of what biology is about. Since there are no fundamental laws of biology, he proposes to organize understanding by scale. Curiously, he does not argue that mathematics might help or be necessary, but he does say that life has to follow from the laws of physics. These lectures were amazingly comprehensive, compact, and they were a clear presentation of virtually all that was known at the time, from biochemistry to immunology. Well, by 1969, the foundations of molecular biology were pretty much laid, and a more predictive, quantitative, theoretical view of biology could have been imagined. And the Hughes lectures give some evidence that Feynman was beginning to think along such lines, but he was hijacked by physics before he could develop such thoughts. Something happened in the late 60s. This was the beginning of the creation of what is known as the standard model of particle physics, how somehow quantum electrodynamics, which he had essentially laid the precise intellectual foundations for in the late 40s, it was transformed into something that explains the strong interactions, the weak interactions, the physics of quarks and leptons and all the rest of that. And he got drawn into that. And he stopped thinking about biology. So I couldn't help but ask myself, how might Feynman's thinking have evolved from there? So theoretical physicists have continued to be fascinated by biology, but the goals of biophysics have evolved since Feynman left the subject years ago. Biology quickly answered the basic molecular questions and began, indeed, to look at how the molecular parts worked together to make a living cell. Leaving aside advances in microscopy, understanding cellular function posed a raft of basic questions that only physical science could answer. Things like, how do bacteria know how to find their food? That has to do with diffusion. How do proteins find their folded state? They emerge from the ribosome as a string of amino acids, and somehow these guys have to fold themselves into some very particular three-dimensional structure. And it ain't obvious that this can happen in a reliable way, and so this is a problem for statistical physics and things like that. Observations of collective cell movement, as in embryonic development, posed questions that were answerable by the new physics of soft condensed matter, and indeed this is a very big subject now. And beyond questions of physics in biology, advances in biology called for a systems approach to understanding how living systems could work. And there are things, well, questions of information transmission and processing, how does evolution work, et cetera, all became questions that you could think about because you could do experiments and get relevant data. These things are not physics in the strict sense, but you can't pursue such matters without building mathematical frameworks like physicists know how to do in pursuing the scientific questions that they have been successful at. So this quest, somehow building the mathematical frameworks that they're not copies of the mathematical frameworks of theoretical physics, but they're inspired by, they have the same goal, create a mathematical framework that can incorporate facts, as it were, and deliver predictive power and understanding. Okay. With some help from Feynman, I will now try to explain this last point. So I collected a Poor Richard's Almanac of Feynmanisms. There are a whole bunch of them. You go through, you must be joking, Mr. Feynman, or any other such, so to say, biographical screed. You'll find him saying things that are quite trenchant and, you know, very, very, how shall I put it, emphatically pronounced. So here are a few of them that I think are useful or plausible jumping off points. It's very easy, and you have to imagine, the other thing about him was when you listen to his lectures, they delivered great clarity and inspiration, but they were delivered in a New York style. You know, you had lots of waving of arms and lots of look at this, and then what you see is you get that. Can you imagine? So you have to think of him saying these things. I tried to find a video clip that had the voice, and I couldn't find one that I really liked, so it's only written. So it's very easy to answer many of these fundamental biological questions. You just look at the thing. Okay? You know why you fellows are making so little progress? You should use more mathematics like we do. I think that was a friendly joke. Biology is not simply writing information, it's doing something about it. Then there's nothing that living things do that cannot be understood from the point of view that they are made of atoms according to the laws of physics. That's an aspirational statement for sure, and in his Hughes lectures, he didn't follow up on that. He said, I don't know how to do this. It's just too complicated. And then finally, this is something which was somewhere in the upper left-hand corner of his last blackboard, probably was something he wrote down and kept there sort of as a continual stimulation. What I cannot create, I do not understand. And I'll come back to that. So these thoughts of a theoretical physicist about biology remain relevant today. They're convenient pegs on which to hang a discussion of what Feynman's followers into this field are finding today as they try to make sense of biology. Okay, you just look at the thing. I won't go too deeply into this. Feynman's visualization challenge was met, and then some, by combining the tools of lasers, sophisticated optics, genetic engineering, and computers. This could be a whole lecture in and unto itself. We can now look inside the cell and can see things beyond the diffraction limit that are on a small enough scale to really directly answer how are these machines, what is the structure of these machines and how do they do what they do? And that is a very, very important development. In short, we can now observe the inner workings of the cell in great detail and in many cases, while they're doing their job. We don't have to kill them in order to look at them in many, many cases. So this is, and you know, it only took 50 years, roughly speaking, to respond to this challenge. You should use more mathematics. Well, that's something I can resonate to. I'm a theoretical physicist. I love mathematics, and that was what drew me to theoretical physics in the first place. So the flip side of our ability to look inside the cell is that the data we can now collect is becoming too copious to directly convey any meaning. The need for mathematical frameworks to make sense of what we see is urgent. So I have some examples here. I won't go through them all, but I mean, everybody understands that sequencing, the ability to look at the actual content, what is in fact written on the DNA of this particular organism. Take one of my cells. We can read in the space of an hour, or less than that. I'm not sure exactly how long it takes. The key point is it's cheap. We can read all 3 billion base pairs in the order in which they are on the chromosomes. And we can do that not just for, you know, a particular species, for many individuals in a species. You could get the level of expressions of thousands of genes under different growth conditions or different points in the cell cycle. We can record, so neurobiologists are having fun in this sense too. We can record what's happening in individual neurons in a brain, not our brain, in a rat brain, or in a retina, not our retina, a salamander retina, but we can record from thousands of neurons in an animal that's alive and doing what it does in its normal course of events. And since neurons can fire every 10 milliseconds or so, and you're recording from thousands of neurons, and you can do this, it turns out, for thousands of seconds before the poor animal gives up, you can gather, you know, in one experimental session, many billions of bits of information, right? And you have to do something with this. It doesn't mean anything by itself. And there's a bunch of other things that go along with this. Biology is, the beneficiary of and the victim of a flood of data, and this flood of data has come from technological developments that are associated with, you know, the miniaturization of computer devices, and also many things that come directly from physics. Okay, so we don't have to do this. Modern physics has a similar big data problem, and I will not go into that because I don't want to run over time, but I think you can believe it. You should use more mathematics. Well, here's where I was going to do a little sidebar in which I would give you an example of what a theoretical physicist think that means. You know, modern biophysics looks for principled theoretical approaches to comprehending biological systems in a way that has some generality. You know, it's not just looking at a very specific organ in a very specific species. It's trying to find things that have to be at work in a wide range of cases. So the basic idea is to apply the theoretical physics of understanding, the theoretical physics method of understanding complex phenomena through the inference of simpler, hidden, mathematically expressible mechanisms. There are several broad threads that I could follow, and I probably, yes, I think I'm just going to have to skip that. I was going to talk about how information and the transmission of information from one stage to another could be developed in understanding how an egg, single cell egg, develops into a full animal with, you know, hundreds of thousands or millions of cells. And this is something that one can do in the context of embryonic development of simple organisms, in this case, the fruit fly. The fruit fly is one of biology's favorite organisms for studying development. I mean, development is truly the most extraordinary phenomenon in biology. You start with a cell. It gets fertilized, and it starts dividing. And then, somehow, as time goes on, as the number of cells increases, structure appears. You begin to see the formation of a head and a tail, and then of legs, and then of wings if you're an insect, or of a gut and and a brain if you're a higher organism, and exactly how that works and how the information that tells this cell, which is here at this stage of an embryo, how does the cell, how does the information that what you should now do is different what you've been doing before, you should do the following different thing, how that is understood, how the quantification of the information that's available to these cells, and is it enough to actually tell it reliably to do what it has to do. This is a kind of approach to understanding development that people coming from theoretical physics find to be an interesting and useful way of trying to engage with biology. And biologists are able nowadays to actually collect data of a kind and quality and quantity that makes it possible to address these questions. So continuing on with the Feynman aphorisms, nothing that living things do cannot be understood from the laws of physics. Well, life is certainly governed by the laws of physics, but how to get from them to the intermediate mathematical principles that we can use in practice is unclear. And basically, to be honest, what we're doing these days, those of us who engage in this kind of exercise, is at best a warm-up exercise for the real problems. The relation to fundamental physical law is remote. There's a menu of less ad hoc questions that we don't know how to attack yet, but that might be conceptually closer to the laws of physics or of statistical mechanics. And, just for an instance, what is it about non-equilibrium statistical mechanics that makes it possible for, starting with just a bath of chemicals, stably reproducing distinct entities can arise? How does this happen? In what way how is this consistent with the laws of physics? It is a question that it is definitely consistent with the laws of physics. We're all convinced of that. But how to get from statistical physics as we understand it today to that level is something which is not known to us. Also, the other thing that should be of great interest to us is brains carry out tasks that have an abstract representation. And can we create models of the network that's represented by, or a network that represents what we have inside our brains or in our sensory system that actually capture the processing power of a human brain or eye. Developing this kind of theory is what theoretical physicists do, and doing it is a challenge for the curious characters who would like to emulate Feynman. So my last aphorism is, what I cannot create, I do not understand. So what he meant by that is something that every person who's become fascinated by physics and has gone to college and studied it and gone to graduate school and learned how to do physics research and tackled new problems, everybody understands this. That is to say, it's kind of part of the culture that when you're faced with a phenomenon, even something that's going on in a coffee cup, you go back to first principles, F equals ma, or something about diffusion or something like that. You know the values of fundamental constants and so on and so on. Can you, more or less on the back of the envelope, or ideally in your head, understand why the liquid is crawling up why the wine is crawling up the side of the glass in these little fingery things? That's the so-called Marangoni effect. Physicists try to connect actual phenomena with first principles, and it's amazing how much you can do. When I was a graduate student, we had to take a very elaborate series of exams. VJ may well have had to do the same thing, and in my day, one of the segments was 3 hours in which you were given a bunch of phenomena, simple phenomena. They're described, and you are to qualitatively or semi-quantitatively explain them, using your understanding of the laws of mechanics and electromagnetism and quantum mechanics. You can explain why the highest mountain on the Earth is 30,000 feet high, knowing the value of h bar and the mass of the electron and the mass of the proton. Okay, you could do this. And physicists love this. This is what he meant by this. He doesn't mean by create, he means create and actually recreate the quantitative argument, the principled argument that explains the fact. And I don't understand unless I have that. And so, he could do that. He wanted to do that for every new thing that was discovered in physics. High temperature, superconductivity, superfluidity, this, that, and the other thing. Okay. And we physicists love this. Now, I fear that this appealing dictum is losing its pertinence. The physicist's ideal of explaining things by pencil and paper reasoning from fundamental law applied to simple abstract models, it already doesn't work in rather large swaths of physics anyway. And the problem arises from emergent phenomena that are not baked into the fundamental equations. And they arise from strong correlations between degrees of freedom that we initially put in by hand. There are many examples of this. Starting with the Schrödinger equation, you ought to be able to derive superconductivity, right? Well, it would have been great if people had predicted superconductivity from the Schrödinger equation when it was invented, but we couldn't do this. If I can't reproduce the existence of, I was mentioning confinement, which is another problem, without the help of massive computation, why should I expect to be able to avoid the same issue in the case of living matter? In the end, it's a question. So we use computation in the sense of you make numerical models and you use big computers. We also used to mean computation, writing down equations and using what we understood to be exact or approximate solutions, not numerical, but functional solutions. It's getting harder and harder to do that second thing. In the end, it's a question of what you mean by understanding in the scientific sense. I don't think the situation for biology is intrinsically any different than it is for, say, nuclear physics. Even mathematicians are beginning to accept computer proofs of complicated theorems, so we probably shouldn't worry about this philosophical issue. Understanding in the sense of I construct some framework and I unfortunately am obliged to submit it to a computational machine and then do something with the outcome. We're used to doing that. Maybe that's okay. We would certainly like to have the ability to understand the cell or evolution or whatever in the Feynman sense. I'm not at all convinced that we're likely to get there, but we should do the work to get as far towards that as we possibly can. Okay, so I think I'm done. Let me just say the problem's a major intellectual challenge. It's just as hard and deep as the fundamental physics challenges we've solved in the past, and accepting this challenge will ensure the future vitality of theoretical physics and biology. And today there are many curious characters from theoretical physics who've been drawn to this challenge, and I'm sure that Feynman, were he with us today, would be attracted to it also. We could certainly use his curiosity and his genius. So thank you very much. I'm happy to take questions, but people who have to, I ran on a little bit, not as much as I usually do, so I'm pretty pleased with myself, but I'm happy to.
I'm so eager to ask you this question, and it may be something that's totally out of your realm, but just from a pedestrian point of view, not being a physicist, I'm wondering if there is a connection. You seem to be a problem solver in your discoveries of why things work and how they work. And how much connection is there between what you do and medical research? For instance, if a protein is misfolding out in a muscle cell, it seems like nature messed up a little bit. Do you tackle problems like that? And if so, how connected are you to medical research?
Well, that's a softball question. No, actually, it turns out the subject that I'm actively interested in is the functioning of the immune system. Yes. Okay? Now, this is actually a problem that has aspects that remind a physicist of what we call statistical physics or statistical mechanics. This is because your immune system consists of lots of T cells and B cells, but the remarkable fact about them is that they're all different. You have let's say, 1 billion different T cells. I mean, different in the sense that each individual T cell has the ability to recognize some particular aspect of a particular pathogen. And we just have lots and lots of different such entities in our body. And we're just, I mean, somehow the system hopes that amongst this billion or so different tools, there will be the right tool to deal with the particular pathogen that shows up. Now, we come to cancer. This is the medical research that I have some connection with. There's this whole field called immunotherapy. People made the observation years ago that, well, the immune system has an important aspect. That is to say, it attacks things that are foreign. It attacks foreign proteins, but you are made of proteins yourself. So the immune system has to somehow train itself not to attack you, but to attack things that are not you, that are sufficiently different from your proteins. So the observation was, well, a cancer cell is a cell that has undergone somewhere in its genome some mutations that mess up the growth control system. So at some level, this cell is foreign. It has inside it a protein that is not one of the proteins that you initially had in your information. Okay? So why doesn't the immune system take care of cancer like that? It just recognizes the foreignness of the tumor cells and kills them. Well, there's a long story because basically, in order to keep the immune system from running out of control, there are breaks. In other words, there are things that cells can do to say to the immune cells, leave me alone, you don't want to go there. And so drugs have been created that in effect turn off this control mechanism, and then the immune system, and it has been demonstrated in some cases, will actually attack the foreign tumor cells. So there's a physics-like question in there, and that is, well, so everybody has a different immune system. That is to say, the number of different kinds of cells that you could have in your body is so large that each of us only has a very sparse sampling of, the whole universe of possible T cells. So there's a question of a kind of theoretical nature for immunotherapy anyway. Given a sort of garden variety cancerous mutation in some protein in a tumor cell, what's the likelihood that a garden variety human being will have in its complement of T cells a T cell that will do the job, actually recognize this particular mutation? So that's the kind of question that I have gotten involved in, and it gets to some very basic statistical, mechanical-like questions about how the immune system is generated and things like that. In principle, who knows, the kind of thing that one finds by following one's physicist nose and studying the way the immune system works as a statistical system could conceivably, I would hope so, have some relevance to actual clinical medicine. And there are many, 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 many examples of this. So the answer is kind of yes.
Yes. It just seems that if the science knows the reason that a disease is happening, a rare disease, for example, if they know it's because the proteins are misfolding,
Oh, well,
why
there's...
can't they fix that?
Well, it's a one-way, misfolding is pretty much a one-way street. I would like to think a little bit more before I try to pursue that path. But I'll go on to one more question.
Physics seems to be expanding its interests. Biology, and now even physicists tend to be showing interest in seeking professions even in Wall Street. Is there any connection to these two or more phenomena of the expansion of interest in other fields by physicists?
Well, okay, so I don't have a complete answer to that, but one thing that you do learn as a physicist is how to construct your stock in trade in some sense is constructing mathematical frameworks that can incorporate facts, data, and do something useful with it. Okay? Now, as everybody knows, there are many, many domains in which facts are being accumulated in vast numbers, and in order to make use of those facts, you need mathematical frameworks. Think of Google, the search algorithm, right? It was not invented by a physicist, but it was invented by mathematicians or computer scientists. But similar issues arise, as you say, in banking and in investment.
So it's not the failure of certain aspects of string theory, for example?
Yeah. No, it's just that, well, we actually, when we talk to undergraduates who are thinking about, shall I major in molecular biology? Shall I major in philosophy? Shall I major in physics? Well, one of the things we say to the youngsters who are interested in majoring in physics is, well, it's hard work, that's for sure, very demanding and all that. On the other hand, the intellectual skills that you develop are applicable in a very direct way, not just in pursuing the classic questions of physics. It is becoming more and more clear that the way the world works, the way our advanced industrial societies work, people who have developed the kind of intellectual skills that training in physics naturally develops, have tools that they can use in fields that are rather remote from physics. And that's what's being, I think that's the phenomenon you're seeing. It's not, how shall I put it? Being a practicing physicist at some level is being a monk. You're giving up the, you're not compensated that richly, except there's tremendous psychic income, as Governor Jerry Brown of California used to say. And some young people want to go out and make money and support their family, and after 10 years come back and be a generous donor or philanthropist. I don't know. It's just, there are many interesting ways you can exercise your skill and your brain. And any others?
Yes.
One more? Yeah.
My question is a little bit aligned with this lady over here. Some of what you're talking about, can it be utilized to predict rate of change, mutations, or anomalies in viruses?
Well, that's, yes. Actually, several of my colleagues have done work that seems to be quite relevant to the question of what flu vaccine should we be, so to say, producing in order to deal with the strain that is going to be most prevalent next year. All right, so there's, that's a kind of meta system. In other words, I talked a little bit about the immune system in an individual human being, but the immune system available to the human beings on the planet is in some degree of competition with the viruses, and in particular the flu virus that attack us. And so I can't go into any of the details, but it is possible by constructing mathematical models that are, they're models, they're kind of the best you can do at the moment, but the idea is in effect to take what you can learn now, maybe what you've learned about what are the strains that are most prevalent this year, the preceding year or two, and from that, using the statistical frameworks that you've developed and crossing your fingers a little bit, predicting what's going to be the most dominant strain next year. They seem to have some success in doing this. It's an expedient. It's not grounded, how shall I put it? If you think about it, trying to talk about how the 5 billion or 7 billion people on the planet who are exposed to the flu virus every year, and thinking about the reservoir of different flu strains that are present on the planet and how these two interact with one another, that is a remarkably complicated problem. And if you could address it in some principled way, getting down to the level that really has to be dealt with if you're going to do a serious theory of it, that would be great. But people are sort of working their way toward that. And as I say, my colleagues who have developed some modeling tools that allow them to make such predictions, apparently they've impressed the World Health Organization enough that they are in the pipeline for making the decision. In other words, they're not the only ones who decide, but their input is sought in the WHO's mechanism for deciding what's the recommended flu vaccine for the next season. So the answer is, yeah, you can hope to do that. It's very complicated. It's like making a model of the entire atmospheric circulation system on the planet in order to predict next week's weather or next century's climate. You can think about doing it. You can do approximations to it. They're not quite good enough yet, but you can hope that with more computing power and more mathematical insight, you can take it to the next level. Thank you all for listening.